Anna Dumitriu and I'm an artist who loves working with bacteria. The Wellcome Collection asked me to suggest an idea for this event, this on-light weekend spectacular that they've been doing. And I love bacteria, I'm fascinated in everything to do with bacteria. And I started to think about the different ways that bacteria and light are related and put together this event, mostly with collaborators and people that I've already worked with or started collaborating with. So it's all related to stories of previous projects that I've done with different people. So this is the Bacteria Light Lab. It's a room full of ways that bacteria either use light, produce light, or can be killed by light, and the ways that new diagnostic techniques are using light as well. Also the history of light and microbiology and the ways that they used to try and cure disease using light, or the way they believed that sunlight would kill tuberculosis and things like that.
I'm Carol Reeves and I'm a senior lecturer in Science and Technology Studies at University College London. Heliotherapy was extremely popular from the 19th century, the idea that sun is very healing, it's very good for you. There was also an idea that you could kill bacteria and in fact ultraviolet light will destroy bacteria such as tuberculosis bacteria. So there were ideas that you had to design buildings like tenement blocks where people could actually get as much light as possible. Then there's the Finsen lamp, and this was developed in the 19th century by a Danish doctor called Niels Finsen. Queen Alexandra, who was the wife of Edward VII of England, she was Danish by birth. She heard about this lamp and she brought the first one back in Britain, and that was placed at the London Hospital in 1900. And immediately that lamp was available for people. There were people queuing up all around the hospital with who had tuberculosis of the skin, which is very disfiguring, and this was seen as a lamp of hope. This was brand new technology when people were lighting their homes with candles. There is some evidence that that can work. We often forget, this is just 100 years ago, that people with these very disfiguring diseases, these very disfiguring facial diseases would be walking around the streets, and this was their hope to be cured.
Yeah, I'm Professor Michael Wilson from University College London, and I work on light-activated antimicrobial agents and also the human microbiota. These are agents that will kill microbes once they've been activated by light of an appropriate wavelength, so they're an alternative to antibiotics. The advantages of light-activated antimicrobial agents over antibiotics are that, first of all, they are effective very, very rapidly, within seconds or minutes, whereas antibiotics take a long time to have an effect. Also, the second main advantage is that it's very unlikely that microbes will develop resistance to these agents, because these agents basically attack many, many targets within the microbial cell, unlike antibiotics, which just target one particular pathway in the cell. You see the blue, methylene blue or rose bengal. These are agents which, if you shine light of the correct wavelength, like for the blue ones, you shine red light on them, and when you shine red light on them, they produce reactive oxygen species such as singlet oxygen, and the singlet oxygen then kills any microbes in the vicinity. So it's a rapid, effective way of killing microbes. I started it about 18 years ago, so I've been working on it for over 18 years, but it's been commercially developed only probably in about the last 10 years.
We're looking at our Winogradsky column beauty contest here. So these are lots of different Winogradsky columns. Sergei Winogradsky was a pioneer of microbiology and a contemporary of Louis Pasteur and Robert Koch. But he was slightly considered a bit of a madman at the time because he believed that what we should be looking at is whole ecosystems of bacteria rather than studying one bacteria in isolation. Which is kind of necessary for medical microbiology in the past, but now with techniques like whole genome sequencing, we can actually start to study how bacteria work as part of a whole ecosystem, and they change their behaviour in different places. So if you've got Staphylococcus aureus living on your body, it may actually behave in different ways and actually change its DNA depending on where it is in the body and how it interacts with you. So his ideas of this kind of whole ecosystem microbiology, which he wrote about in his famous text, The Microbiology of Soil, is now becoming a sort of current paradigm for microbiology, sort of 130 years after he kind of talked about it in the first place. So he invented this method of culturing sulfur bacteria in these kind of methods called Winogradsky columns, where you put in different layers of stuff, and we've been doing workshops here at the Wellcome Collection over the past few days so people can make their own. And you use salt marsh mud, egg yolk, eggshell, plaster of Paris, tree bark, torn up newspaper, topsoil and water. And then you put the lid on and you leave it, and the bacteria that are in the salt marsh mud start to do their thing. And over time they create these amazing ecosystems where, depending on how much oxygen is getting down there or how much light is getting to it, because they do react to the light, you create these wonderful layered ecosystems. And you can kind of create images where if the light is prevented from getting to part of the column, you'll grow things like the purple bacteria that like more darkness. So you can put them in different kinds of light, and it will change in different ways. These two that Simon Parkes made, one was kept in the dark and this one was kept in the light. So you can see that really affects them as well. So pretty much everything you do to them will personalise them in some way. It's a really complex ecosystem, so it changes and changes with the slightest sort of differences. This one has lots of different cyanobacteria in it, and this beautiful green that it's creating. You can keep them with you for your whole life. They don't take much care, and they're kind of work as kind of amazing pets really as well.
And I'm Nicola Fawcett, so I'm actually usually a frontline doctor, but I'm doing a research project with the Modernising Medical Microbiology Consortium looking at antibiotic resistance.
We're looking at a video that we've put together of how light is used in whole genome sequencing of bacteria, this brand new technology. It's a very important new area for infection control and science and healthcare, and it's going to affect how people's healthcare is undertaken in the future.
So actually what you're seeing here, the flashing, is essentially a forest of DNA strands. And this is what the machine sees, sequencing a tuberculosis bacterial DNA sequence. So each flash is a group of highly similar DNA strands producing a fluorescent light when a laser is shone on it. The machine essentially takes digital pictures like we would with a camera. It remembers it saw an A, then a T, then a C.
And it pieces them back together because each fragment of DNA is also tagged with a kind of DNA barcode as well. So they can tell that this bit joins up to this bit, joins up to this bit. And then the bioinformaticians then work on the data to actually do this jigsaw puzzle. So you can actually learn all about its drug resistances, and in the future we hopefully will be able to give the sort of the full diagnosis and kind of work out what the treatment for a condition is just from looking at the whole genome.
What's incredible incredible is the amount of information that we get from one sequence run. So actually the amount of information that is coming off what you see in front of you is about 20 tuberculosis bacterial genomes in multiple copies, all being sequenced at once. And a way of explaining just how vast it is one bacterial genome from tuberculosis, if I read it out at A, T, C, G, would probably take me about 23 days to read out. And the amount of information right here would probably take me about 50 years. Now what this is effectively doing is asking 15 million people, or roughly the population of the Netherlands, to all read it out at the same time in 2.5 minute chunks, and then trying to do the world's most complicated jigsaw and put it all back together. And so what you're seeing is, if you like, 15 million flashes as it's being read out.
I'm Dr. Simon Park, and I'm a microbiologist from the University of Surrey. Basically, you've got old laboratory equipment filled with artificial seawater agar, and it's been inoculated with a bacterium called Photobacterium phosphorium that lives naturally in seawater, and when it grows it naturally produces this lovely blue-green glow that you can see before you. One of the really fascinating things about the light that you see here is that when these organisms are actually in association with the fish or the anglerfish, they produce the light, but when they're free living in seawater they don't, because the light that they produce is incredibly energetically expensive, so they'd much rather not do it if they don't have to. So when people first started to study bioluminescence, they wanted to know what the switch was that the bacteria in the fish recognised that the bacteria in the general environment didn't recognise, that told them to produce light in the fish. And after about 5 or 10 years of research, they found it wasn't a switch that the fish had, but it was the bacteria actually communicating with each other. So when they're in the light organ of a fish, they produce a chemical that builds up to a certain threshold, and when that builds up to a threshold, they know there are enough bacteria around them that if they start producing light, it will produce light that will be able to be seen. But when they're in seawater as single cells, they still produce that chemical, but it never builds up to a high enough concentration. So that was the first study really that showed bacteria talked to each other. And that was in this exotic bacterium, but now we know 20, 30 years later that all bacteria talk to each other, and to cause infection they have to talk to each other, and to do many of the things they do in the environment they also have to talk to each other. So these sort of studies in something quite exotic has led to something quite fundamental in terms of microbiology.
And understand how they communicate, and you could maybe tell them to do things other than what they're doing. The lady who first discovered it, who works with Bonnie Basler, who works with Vibrio fischeri bacteria, she's managed to block the receptors in the Vibrio fischeri bacteria to stop them glowing. And that doesn't sound very exciting until you think that Vibrio fischeri is very related to another Vibrio bacteria, Vibrio cholerae, which uses quorum sensing to turn on toxin production, which is the thing that makes people really ill. So if you could stop that using this method, it could be a very useful new antibiotic treatment. The work with the Modernising Medical Microbiology project is part of my ongoing work to investigate how whole genome sequencing works and engage the public in it, and also create artworks that investigate it. So I'm trying to hands-on understand it by practically working with it and interrogating it as an artist from an artistic paradigm to create a new installation. And also it's that idea of people can stand and they can talk to the scientists and have a chat and really ask questions in a really non-hierarchical way, just one-to-one conversations and chats, and it really breaks down the barriers there. So normally people don't just get to chat to scientists, and that's what I really like, is that sort of breaking down the barriers. It's funny because the artists in the room are wearing lab coats, especially made bacteria light lab coats, but the scientists aren't wearing lab coats. That's how you can tell we're artists, is we're wearing a lab coat today.